Hello everyone, welcome to our webinar entitled The Human Microbiome, Its Impact on Our Lives and Our Health. Our guest speaker today is Dr. Robert Rountree. My name is Christine Stubbe and I am a medical education specialist at Genova's Asheville branch. I am going to serve as the moderator for today's webinar. We would like to welcome Dr. Robert Rountree. Dr. Rountree has been providing a unique combination of traditional family medicine, nutrition, medical herbology, and mind-body therapy in Boulder, Colorado since 1983. In addition to writing numerous book chapters and magazine articles, he is co-author of 5 books on integrative and nutritional medicine: The New Breastfeeding Diet Plan, Smart Medicine for a Healthier Child, A Natural Guide to Pregnancy and Postpartum Health, Immunotics, a revolutionary way to fight infection, Beat Chronic Illness and Stay Well, and A Parent's Guide to Medical Emergencies. He is the medical editor for Delicious Living and serves as a clinical editor and regular columnist for the journal Alternative and Complementary Therapies. He currently serves as the Chief Medical Officer of Thorne Research and is a member of the Scientific Advisory Board for Albion Laboratories. He is a long-standing member of the adjunct faculty for the Institute for Functional Medicine in Federal Way, Washington, as well as the Fellowship in Anti-Aging and Regenerative Medicine, sponsored by the American Academy of Anti-Aging Medicine. He has lectured to professional and public audiences all over the world. In addition to his professional pursuits, Dr. Rountree is passionate about the outdoors, wild nature, and the preservation of biodiversity. So now I am going to turn the control over to Dr. Rountree, and I want to thank everybody for being here today.
Terrific. Well, hello everybody. Thank you for tuning in. This is definitely one of my favorite topics to discuss, and it's an area where the research has just been exploding in the last few years. It seems like everyone is suddenly talking about the human microbiome. It almost started with an editorial that Michael Pollan wrote in the New York Times a couple of years ago entitled, "Some of My Best Friends Are Germs." And that gives you an idea of where the thinking has shifted in terms of the microbiome. So what we're going to talk about in the next 45 minutes are some of these new discoveries. Number one, how the use of DNA sequencing to determine what's really there in the gut or on the skin or in the mouth has told an entirely different story than what we used to think in terms of what was actually there. This DNA sequencing technique has really redefined what it means to be human. I'm going to talk a little bit about how the gut microbiome affects metabolism, neurology, immunology, to show how our modern lifestyle and our diet in particular has an impact on the microbiome and how that affects chronic diseases. And then finally, a few comments about how things that we can do, like prebiotics or probiotics, can help maintain a healthy microbiome. So here we have a picture of gut bacteria lining the gut mucosa, just to give you an idea that we are covered with microbes of all types. I like this analogy that human beings are basically like coral reefs. They're these very complex ecosystems that cover our outer surfaces and our inner surfaces. And they're vast microbial ecosystems that are involved in these different surfaces. Now, every individual has what appears to be a kind of core microbiome that doesn't really change from day to day. It may change briefly. You could, say, swab a person's arm with some alcohol and temporarily disinfect that part of the arm, but within a few hours of doing that swabbing, basically the same bacteria and viruses and fungi will repopulate. So there appears to be a unique microbial fingerprint that every individual has. And in the future, many people think that that fingerprint may be used in the same way that we currently use fingerprints to determine whether somebody has been in a room or touched a piece of an object or has been present in a certain area, that this fingerprint is that unique that we can actually tell if somebody has been in a specific environment. So what kind of microbiota are we talking about? Well, viruses in particular, acellular forms. There's a whole study of the human virome, and in particular it looks at bacteriophages, and the concern about bacteriophages is that they can carry genes, what's called mobile genetic elements, back and forth to different microbial environments. So this is a concern when it comes to antibiotic resistance. Then we have prokaryotes, which also have no nucleus, and there are two major groups here: Archaea, which are strict anaerobes, and we'll talk about Archaea in a little bit of detail, and then bacteria, which are aerobes and anaerobes. And then finally, part of the microbiota includes microfungi, protozoa, and even parasitic worms. And all of these form a soup, a mix, which is interacting with the gut mucosa, which is interacting with the gut-associated lymphoid tissue to have an impact on our overall physiology and immunology. So let's talk a little bit about Archaea. It was thought for a long time that Archaea only lived in very extreme environments, super cold environments like the North Pole or hot springs, places where nothing else could really live. Now we know Archaea can actually live in the human body. They've been found in the mouth, and they've been found in the gut. They're strict anaerobes. They're very difficult to culture. And so the use of DNA sequencing, using PCR to sequence DNA, has actually allowed us to determine the presence of Archaea in the human gut. They can also be determined by breath testing. Initially, it was thought that they were commensals, that they were all our friends, but now there's a number of studies showing that overgrowth of Archaea have been implicated in a range of both oral and GI disorders. Oral disorders such as periodontal disease, and GI disorders like chronic constipation or SIBO. This is an article by Dr. Pimentel, who I believe is at Cedars-Sinai and is one of the world's experts in SIBO. It's a review article on Methanogens, and he's specifically talking about methanogenic Archaea being linked to constipation and irritable bowel syndrome. It turns out that methane that's produced by these Archaea has an impact on intestinal transit and pH. So if there's a lot of methane being produced, it slows down intestinal transit time and facilitates development of constipation. The other thing methanogens can do is they help break down carbohydrates more thoroughly, which leads to a higher production and absorption of short-chain fatty acids. In some ways, this could be beneficial, but if you consider that this implies a higher level of extraction of energy from food, it could be linked to obesity. Now, this is very new science, so I wouldn't say that that you should see this engraved in stone yet. But these are some of the questions that are being raised about what these Archaea may be doing inside of our bodies. The main Archaea of concern is Methanobrevibacter smithii. It's the dominant Archaea in the GI tract. About 10% of prokaryotes in the gut are Archaea. However, about half of the people tested don't have any at all, so it's not considered a standard commensal. As I said, it produces methane either from hydrogen or carbon dioxide or short-chain fatty acids. And this is actually a major source of flatulence in cows, and believe it or not, there's articles suggesting that these methanogens may be contributing to global warming. As I mentioned, Dr. Pimentel's work has implicated Methanobrevibacter smithii in constipation-predominant irritable bowel syndrome and small intestinal bacterial overgrowth, or we should actually call that small intestinal microbial overgrowth, in obesity, and even in type 2 diabetes. Again, if you're extracting more energy from food, you're more likely to become obese, and you're more likely to have higher blood sugars after eating a standard meal. it has been pointed out that methane breath tests are a good surrogate marker for the presence of methanogenic archaea. And in fact, this is probably a better way to determine small intestinal microbial overgrowth with methanogens than doing a stool analysis, since with a stool analysis we're looking at what's in the colon and not in the small intestine. So let's just look at the big picture when we're talking about bacteria. In the skin, we have about 1 trillion resident bacteria. In the mouth, about 10 billion. In the gut, about 100 trillion bacteria that weigh about 2 to 5 pounds, depending on the individual. And most of those bacteria are anaerobes. It's been estimated, based on this new DNA typing, DNA sequencing, that there can be up to 1,000 different species that inhabit the oral and gut surfaces. However, even though there's a wide range of variation here, about 99% of us are only populated by 30 to 40 species. An important concept here is that of biodiversity. The total number of species that a person has is called the species richness. And then the amount of each one of these species is called the relative species abundance. These are terms that are used in the biological literature, and both of them are really synonymous with overall health. The more species richness a person has, the more relative species abundance a person has, generally the healthier they are. The dominant phyla that we're talking about in mucosal tissues and in the skin are Bacteroidetes, Firmicutes, Actinobacteria, and Proteobacteria. And you will notice if you do the GIFX profile from Genova that all of these phyla are represented, and there are individual members of these phyla that are actually counted through DNA analysis. So what kind of weights are we talking about here? In the nose, about 10 grams of microbes. And the main resident in the nose is Streptococcus. In the mouth, twice that, about 20 grams. In the vaginal area, 20 grams, about the same that's in the mouth. And in the skin, 200 grams, roughly about half a pound of microbes. A typical resident is Staphylococcus. The total mass of microbes in the gut is about 1,000 grams. As I said, anywhere from 2 to 5 pounds, and Bacteroides is a very typical resident. The new research has shown us that we are outnumbered by bacteria approximately 10 to 1. So there's about 10 times more microbial cells than there are human cells. This is obviously a very, very rough estimate, but it does give us a sense that there are more of them than there are of us. Human somatic and germ cells, about 10 trillion. Total microbiota, at least 100 trillion. So that's a 10 to 1. And if you look at the complexity of this ecosystem, it is actually one of the most complex ecosystems on the entire planet. Looking at soil, looking at rainforest, the human gut really beats them all. So where do we get this human microbiome from? Well, the initial inoculum comes during pregnancy. It was thought that the womb was sterile, that amniotic fluid was sterile. Now we know that there are actually a few bacteria there. Not necessarily those that would come out with culturing, but when you do the DNA sequencing, that's when you find them. If the baby is born by a normal spontaneous vaginal delivery, then the microbiome pretty much develops along the same lines as the species that are present in the mother's vagina. And in contrast, if the baby is born by C-section, then the baby's microbiome will develop along the lines of the skin flora of the mother and the hospital attendants and the bacteria that are in the room where the birth took place. And this has some major implications for long-term health of the baby. In addition, if the baby is breastfed, then there's bacteria that are found in the breast milk that are actually identical to what's in the mother's GI tract. This is pretty fascinating. It turns out that there are dendritic cells in the mother's GI tract that will engulf the bacteria and transport them into the breast milk. So this is a deliberate way of seeding the baby's gut through the breast milk. We used to think breast milk is sterile, and now we know that there's this deliberate seeding that goes on. So the microbes we acquire at birth will affect our health throughout our entire lives. We are born 99% human, but by the time we die, we are 90% microbial. This is a very profound implication. And it says a lot about childbirth practices and what we need to be aware of how we raise infants in the first year of life, because that baby's gut microbiome can really get set, almost engraved in stone in the first year in life, and that can have an effect for years afterwards. If a baby is born by cesarean section, it's been shown that that increases the child's risk of having asthma, allergic rhinitis, celiac disease. In other words, the kid could have the genes for celiac disease but may not develop it. But if they're born by cesarean section, the chance of having the phenotype of celiac disease goes way up. Also, type 1 diabetes and inflammatory bowel disease, all as a result of being born by C-section and presumably being seeded with an entirely different microbiome at birth. One thing that's really fascinating to me is how the microbiomes basically are determined by the microecology of a body habitat. So under the arms are like a rainforest. On the surface of the hand, it's more like a desert. The oral cavity obviously is going to be continuously wet. So that's going to be like the environment you would have under a waterfall. So every one of these microecological zones is going to determine the type of bacteria, the type of fungi, the type of viruses that will live there. Even so, every individual is going to have their own unique composition, as I said, and this is equivalent for crime purposes, you might say. It's doing their own fingerprint. We can actually prove that an individual has been in a room based on the uniqueness of their microbiome. These things do change a little bit over time. They change every time you wash your hands, but they tend to bounce back to a norm every time some kind of intervention has occurred. If you look at the mouth, there's about 6 to 10 billion bacteria, as I've mentioned. There's also archaea, protozoa, fungi, and viruses. Now, this is an interesting study that was published in the journal Microbiome. Yes, there is a journal entitled Microbiome. This was published just last year, and it was a study where they typed bacteria in couples. And what they found was that kissing for about 10 seconds transfers an average of 80 million bacteria. And couples that kiss more frequently and longer have much more similar microbiota. And where they found most of the similarity was on the surface of the tongue. The implication of that is that when people live together, not just couples but families, they end up sharing much of the same microbiota. The same germs will move from person to person. And even if they have pets, if they have dogs, it's been shown that they will share some of the microbiota with their pets. As far as the oral microbiota, for those of you who are dentists or have an interest in this area, there's about 50% of our bacteria that have not been either cultivated or phenotypically characterized. So there's a lot going on in there that we don't yet know about. And the comment has been made that we used to think that periodontitis was a result of overgrowth of a particular pathogen. Well, there may be some disease-associated bacterial species, some pathogens, but probably what's mostly going on with periodontitis is that it's an inappropriate inflammatory reaction to normal microbiota, which is a very similar phenomenon to what we're talking about with inflammatory bowel diseases like Crohn's disease. So let's go on to the GI tract. Again, 100 trillion bacteria in the human gut. In the stomach, we used to think the stomach was completely sterile, and if somebody had Helicobacter, then that was an abnormal situation that would lead to ulcers. Now we know that Helicobacter is actually a normal denizen of the stomach, that it's normal for many, many people to have Helicobacter, which is very important. Just because you find Helicobacter in a stool analysis doesn't mean that it has to be treated. Other bacteria in the stomach, streptococci, lactobacilli, staphylococci. Then the small intestine has roughly the same number of bacteria that you have in the stomach, but you begin to see more anaerobes than facultative anaerobics. Then when you get closer to the ileocecal valve, that's where the anaerobes really start to go up, and that's where you see the Clostridium, the Bacteroides, the Fusobacterium. By the time you get way down in the colon, almost all the bacteria, very strict anaerobes, and this is where you start seeing the really large numbers of bacteria per gram, 10 to 10 bacteria per gram. By the time you get to the stool, 10 to the 12th to the 13th bacteria per gram in the stool. The cell density in the colon are the highest known cell densities for any known ecosystem, according to the Genome Institute at Washington University. So let's talk a little bit about SIBO, or as I said, maybe it's more appropriately called small intestinal microbial overgrowth, because Archaea are not technically bacteria. They look like bacteria, but they have different chemistries. Dr. Pimentel's hypothesis is that small intestinal microbial overgrowth results when Archaea and coliforms like E. coli and Klebsiella expand from the colon backwards into the small intestine. And what allows this to occur? Well, it can occur for people who are taking proton pump inhibitors that don't have enough stomach acid that will help neutralize those bacteria. It happens when the pancreas is not working properly. There's not enough enzymes around, which also will help neutralize those bacteria, keep them from overgrowing. It happens with immunodeficiency syndromes. It happens with anatomical abnormalities and with motility disorders. And many of you who've been following this research may know that Dr. Pimentel believes that anyone with irritable bowel syndrome potentially has some degree of small intestinal microbial overgrowth. His belief is that this should always be treated with antibiotics, but I can't say I'm so sure about that, because that can introduce its own problems, especially long-term dysbiosis. So as I said, we get colonized in the first year of life, either from the mother's vaginal microbiome or from the skin of a mother or hospital co-workers. But over the next 1 to 2.5 years, there's this gradual formation in the microbiome that then gradually gets predominantly colonized by Firmicutes and Bacteroidetes, which is about 90% of the species found. A very profound discovery from studying the genes made by these bacteria is that they have about anywhere from 5 to 10 million unique genes. And humans have about 20,000 genes. So many, many more bacterial and archaeal and viral and fungal genes are found in our gut and on our skin than are in our somatic cells. And the other profound discovery that's come out of this is that that DNA is mobile. In fact, the term used to talk about this is mobile genetic elements. Largely through bacteriophages, this DNA is being moved back and forth all the time. So this has some pretty significant implications for overall health. Human beings have really evolved their microbiome over millions of years. Humans haven't been here for millions of years, but our predecessors have been, and it's thought that we've basically inherited some parts of our microbiome from our predecessors ever since this symbiotic relationship got started with eukaryotes and prokaryotes many millions and millions of years ago. So the human metagenome, which refers to the combination of the human genes and all the microbial genes is really an amalgamation of all these genes. What we see as genetic expression is not just the 20,000 human genes. This is really, really powerful discovery. Now, one thing I should say is even though we're talking about 3 to 10 million potential genes to be expressed, a staggering diversity, a lot of them perform very similar functions in different individuals. So there's quite a bit of overlap in the gene. And let's talk a bit about what some of the functions of these genes are. Well, I've already mentioned digestion and energy harvesting, extracting energy from food, detoxification and biotransformation. It's now thought that up to 50% of all the biotransformation of xenobiotics, hormones, toxins, any foreign materials that we ingest, about 50% of that takes place in the gut bacteria, which means that a lot of the old theories about an individual's genes, the SNPs that they have that impact biotransformation, only play a partial role in how they metabolize xenobiotics or drugs. Our gut microflora also make short-chain fatty acids, they make amino acids, they make neurotransmitters. And you can see the implications of that. If there's an imbalance, a dysbiosis, and those gut bacteria are not making enough of the right kind of neurotransmitter, like they're not making enough serotonin, that can affect mood and behavior. They make vitamins. If you wipe out the gut bacteria, you can develop a deficiency of vitamin K, for example. The gut bacteria line the mucosa and actually prevent pathogens from occupying those binding sites. And this is called humorously colonization resistance against pathogens, or CRAP. And this is actually a term that appears in the medical literature. And then finally, the gut microbiome is involved in systemic immune modulation. It's quite clear that the whole immune system of the body is regulated by the gut microbiota. And specifically, it's the commensal bacteria, the Bifidobacterium, Bacteroides, Clostridium, Lactobacillus, are all associated with setting the overall tone of the immune response. Is the overall immune response one of an inflammatory nature? Is it an autoimmune response? Or is it protection against autoimmunity? Is it a tolerant response? This all seems to be determined by the specific types of commensal bacteria. Again, as Michael Pollan said, some of my best friends are germs, and those best friends are the ones that can determine whether you have a quiet, calm, happy immune system or one that's constantly inflamed. Let's talk about one bacteria in particular, and this is a bacteria that is analyzed in the GIFX panel. It's Faecalibacterium prausnitzii. A number of studies have been written about this. There was a review article, I think, in the most recent issue of Scientific American. It is the most abundant bacterium in the intestinal microbiota of healthy adults. I want to stress that, healthy adults. So in a healthy adult, up to 15% of the total bacteria population is Faecalibacterium prausnitzii. It makes butyrate, and we all know that butyrate is a very healthy chemical for the gut mucosa, especially in the lower part of the intestine. And it has some fairly profound anti-inflammatory mucosal protective properties. This is why several studies have shown that people with Crohn's disease and other inflammatory bowel disease, ulcerative colitis, often have a deficiency of Faecalibacterium prausnitzii. And here's a question to be asked. Is this why some people benefit from fecal transplants? Because they're getting this particular bacteria in those fecal transplants. As far as I know, it's not available in any probiotics available right now. This bacteria is very, very sensitive to oxidative stress. So if a person has a lot of free radicals in their system, it can kill it off, and that, again, can make them prone to inflammatory disease. So looking at the relative abundance of this one bacteria can serve as a biomarker for overall intestinal health, and it's especially useful for Crohn's disease and possibly ulcerative colitis as well. So what are the major influences on our individual microbiomes? Well, our genes, to some extent. This is an area that needs to be explored a lot because we don't completely understand it. But our genes will determine which bacteria are allowed to colonize. The type of birth I've already mentioned. Excessive hygiene. It turns out that a lot of the bacteria that colonize our gut actually come from our food. Yes, we are getting live bacteria from our food. If you eat salads, no matter how much you wash your vegetables and your fruits, you're going to get some bacteria from that. And if you look at hunter-gatherer tribes who are perhaps less particular about their hygiene, they have a much more rich microbiome, and a lot of that comes from the food that they eat. So being excessively clean actually can lead to a loss of richness of the gut bacteria. Our diet has a fairly profound effect, and an effect that can be exerted within a couple of days. And then eating probiotics and prebiotics, fibers that work as fertilizer to help probiotics grow. Medications, antibiotics in particular, have a profound effect on the microbiome. Non-steroidal anti-inflammatory drugs. Social and emotional stress. A number of animal studies have shown that the microbiome can change quickly after social stresses. Any kind of chronic disease impacts the microbiome. Aging leads to dysbiosis and a decrease in richness. Surgery, and then obviously fecal bacteriotherapy transplants can completely change a person's microbiome. This was an interesting new study that compared the microbiomes of people that ate an all-animal diet and an all-plant diet. So they took 9 volunteers. They had them on 2 extreme diets for 5 days. One diet, meat, egg, and cheese. The other one, grains, vegetables, and legumes. What they found was that when they did the 16S ribosomal RNA sequencing, which looks at the DNA sequencing in bacteria, within 1 to 2 days, major alterations in the microbiome. And the alterations had to do with the kind of bacteria that break down carbohydrates versus the kind of bacteria that break down animal protein. So the people that were on the animal diet had an increased abundance of bile-tolerant organisms like Phyla wadsworthia, which incidentally has been linked to colitis. They had a decrease in Firmicutes that metabolizes plant polysaccharides, and exactly the opposite occurred in those who ate the carbohydrate diet. They had an increase in Firmicutes. The other thing that they found, which I've already mentioned, is that they were able to show that food-borne microbes that were found in both diets colonized the gut transiently, and that included bacteria, fungi, and viruses. So we are getting microbes from our diet all of the time. The big concern here is dysbiosis and reduced microbial diversity. And this is an excerpt from the GIFX profile. I find this to be a very useful marker for a person's overall health. If you look on the left, the diversity association, this person has a moderately high diversity association. I would say that so far every patient that I've tested now that had any kind of chronic GI problem is very low on the diversity association index. So this is a very good overall marker for dysbiosis and for the general health of the person's colonic microbes. And you see on the right the relative abundance of various phyla can be very useful to compare to a healthy cohort. So as I've mentioned several times, a lack of microbial species richness and diversity is associated with poor health. This is emerging as kind of being the marker to look for. Not so much looking for individual pathogens. That was the old way of doing things. Now we're looking at the overall big picture, the systems biology. And we know that very sterile environments, diets low in plant fiber, exposure to antibiotics, all these things decrease microbial diversity. So that's really the target that we want to go after. Who's got great microbial diversity? Hunter-gatherers like the Hadza in Tanzania. There's been some very elegant studies done on them through the Human Microbiome Project that have shown that they have some of the richest microbial diversity of any population on the planet. Dr. Martin Blaser, who's chair of Department of Medicine, New York University Langone, has written this book called Missing Microbes. And Dr. Blaser has a pretty robust set of data, I think, backing his concerns that we are killing our beneficial bacteria and it's causing a number of health problems. He says that, as I mentioned earlier, our microbiome has really evolved over 1 billion years. He reiterates the notion that babies get their microbiome from their mothers at birth, and that every baby that's born by cesarean section is going to lose a little bit of the richness of that microbiome that's been present for a long time. Now we know that even one course of antibiotics can cause a disruption in the microbiome that some people never completely recover from. And that's why he believes that overuse of antibiotics is fueling the increase in chronic diseases like obesity, type 1 diabetes, inflammatory bowel disease, allergies, and asthma. These diseases are all going up dramatically in our population. So this is kind of the next step up of the hygiene hypothesis. Not only are we too clean, but we're blasting out our healthy bacteria. We're killing them regularly. This is one of many studies showing that reduced diversity is a marker that's been associated with increased disease. Reduced diversity of intestinal microbiota during infancy associated with increased risk of allergies at school age. There are a number of different studies that are showing this. This is also a study that came out last year of people who were staying in the ICU who were found to have a complete ecological collapse. These people end up with only up to 1 to 4 different bacteria, most of which are pathogens. And all of them had overgrowth of Candida albicans and Candida glabrata. Now, the very interesting thing was these people weren't necessarily getting sick because even though there's a small number of pathogens, those pathogens are all keeping each other in check. But all it takes is one stressful event. Even putting a person on an opioid can create an imbalance which allows one of those pathogens to suddenly flare up, get out of control, lead to sepsis, and actually can be life-threatening. So this makes a pretty strong case, I think, for doing DNA stool analysis on every patient that's in an ICU and probably every patient that's in the hospital. Here are all the different things that can lead to dysbiosis: genetics, diet, stress, other lifestyle issues, being born in a hospital, vaccinations, antibiotics, excessive hygiene can all lead to disruption in a healthy gut microbiome. And what has that disruption been associated with? A wide range of diseases, acne vulgaris, allergies, asthma, a number of autoimmune diseases, diabetes, reactive arthritis, RA, ankylosing spondylitis, autism, cancer, especially colorectal cancer, a wide range of cardiovascular diseases, not just atherosclerosis, irritable bowel syndrome, Crohn's and ulcerative colitis, fatty liver, obesity, and aging in general. So I wanted to bring up this issue of fecal bacteria therapy, which has really gotten to be quite popular. As it turns out, there's a number of companies that are now starting to provide pooled donor sources of fecal material to be used as transplants. So this is, in the last 2 years, I would say, this whole area has really exploded. Well, what's going on? Why does this work so well for Clostridium difficile? Well, I think it's because we're transplanting an ecosystem. I think this is really different than giving somebody Lactobacillus acidophilus. Probiotics are fine. I think prebiotics can be very helpful. I certainly prescribe them in my medical practice, but we're learning something about what the gut needs to really get restored from the effectiveness of fecal bacteria therapy. Hopefully we'll someday reach a point where we can we can transfer just the needed bacteria and not have to do the whole sample, which will get past the yuck factor. And there are several studies going on now that are trying to determine, is it the Faecalibacterium, Bacterium prausnitzii, or other bacteria like that that make the difference. So what I've told you in the last 45 minutes is that using DNA sequencing to help find out what's really there in our skin and our nose and our mouth and our guts has really redefined what it means to be human. And what that means is that we are superorganisms. We are an amalgamation, a blend of genetic traits from our own somatic cells and the microbial cells that are populating us. We know that our microbiomes can impact our digestion, metabolism, our neurology, and our immunology. And immunology in particularly is impacted when it comes to autoimmune disease or chronic inflammatory diseases. It may be that the presence of a single bacteria is all it takes to set off a major inflammatory cascade. Or maybe it's an imbalance. We don't yet know. What we do know is that the vast majority of our interactions are friendly with bacteria. Good fences make good neighbors. As long as our intestinal mucosa is intact and those healthy bacteria don't get to stream into our lymphatic system or our bloodstream, we can get along just fine. Not only do we get along, but we have a mutually beneficial relationship. We also know that our modern lifestyle, which combines poor diets with lots of refined foods, low fiber, excessively sterile, high stress environments, and overuse of antibiotics, not just the prescription ones, but the antimicrobial substances that are in our soaps, that are in plastics, can be responsible for an epidemic of chronic diseases. And what can we do now to maintain a healthy microbiome? Well, more and more research is coming out pointing to the kinds of diets that are very helpful. A wide diversity of diet that includes a mixture of fibers in plant foods and some animal foods, along with prebiotics and probiotics, are probably the best way to maintain a healthy microbiome overall. So now would be the time to open it up for questions. I hope that you all have learned something and that I've stimulated your thinking, and would be happy to talk to you for a few minutes.
Wonderful. Thank you so much, Dr. Rountree. That was really a great presentation. It's such a fascinating and very important topic. I just wanted to remind everyone that the PowerPoint will be available on our website in a PDF format next week, so you'll be able to listen to it again next week. And so we did receive quite a few clinical questions. One of them was about babies who are born by C-section. What should people be doing to inoculate the baby with appropriate bacteria at that point?
Well, there are a couple of commercial products that are available that are designed just for babies. So I can just say that there are commercial products. They're not hard to find. They're people that have booths at the various functional medicine conferences. And one in particular I know is, so bifidobacteria is what you usually give to babies, and typically what I have mothers do is stick their finger in their mouth, get it a little wet, and then dip it in the probiotic powder and then put that in the baby's mouth, or they can put it right around the nipple when they're breastfeeding. And I think this is a very safe practice, and I think it potentially can make a really big difference. Obviously, we'd like to see some long-term studies. There are some studies using Lactobacillus GG. Even though we tend to think that bifidobacteria is the way to go, that particular bacteria has got several published studies on it, giving it to infants, showing that it can lead to fewer allergies with aging. so, this is not just theoretical. There actually is some published research behind it.
Okay, great. Another question about treating H. pylori. So what determines if you treat or not? Do you go by the amount of H. pylori or severity of symptoms?
It's strictly symptoms, I think.
Mm-hmm.
I think, like most practitioners, when we got the ability to test more readily for H. pylori, I assumed that everybody who tested positive needed to be treated, and so I treated everybody. But then I started reading Dr. Blazer's work, and Dr. Blazer has really cautioned us about not doing that. He basically says if the person is asymptomatic, there's no need to treat. So it doesn't matter what the numbers are.
Mm-hmm.
So clearly, if a person's got an ulcer, you've got to treat it, because it's not even necessarily that the H. pylori caused the ulcer, but it seems to perpetuate the ulcer.
Mm-hmm.
So you have to treat it to allow the ulcer to heal. So if the person has significant dyspepsia, or a proven peptic ulcer disease, then again, I treat it, and I usually just do the standard triple antibiotic regimen. But then be aware that you're going to cause a dysbiosis with that, and then you really need to load the person up with probiotics afterwards.
Mm-hmm. Okay.
What this means, though, is that your goal, the treatment goal is to get rid of the ulcer. The treatment goal is not to completely eradicate H. pylori forever.
Right. Okay, thank you. And then a couple questions about treating SIBO. So if you're not treating with antibiotics, how would you treat it naturally?
Well, Dr. Gerald Mullen, who's at Johns Hopkins, is a functional medicine gastroenterologist, actually published a very nice study on using herbal antimicrobials to treat SIBO. I can't tell you exactly what his regimen was, but I think at least one of the ingredients was an oregano oil. But I followed suit, and I'm much more likely to use a combination of betaine hydrochloride, again, because I think hydrochloric acid helps clean out the small intestine, pancreatic enzymes, and then I use an antimicrobial formula. And, there's a number of them out there. Oregano oil, berberine is another really good one, garlic. So I find that the combination is better than any one individually. The downside to using the antibiotics is, first of all, the one that's used the most often, rifaximin, is pretty expensive. And after talking to Alessio Fasano, who's the celiac, great celiac researcher, he says that when you go in with antibiotics, it's basically like using a bomb to knock out gang warfare. So you've got a localized problem, and then you're dropping an atomic bomb, and it's just blasting everything, and you can't create problems that may persist over the long term.
Mm-hmm. All right, thank you. Another question here. How much does the production of neurotransmitters produced by the bacteria account for the total concentration in the body, and can it cross the blood-brain barrier?
Well, first of all, we know that some 90% of the serotonin in the body is produced in the gut.
Mm-hmm.
Okay? How much of that is from bacteria and how much of it is for neurons is anybody's guess.
Mm-hmm.
Right? So it's, we're superorganisms. So it's a team. And so all I can tell you is that most of it's coming from the gut. How much of that is crossing the blood-brain? I don't know how you'd be able to tell that. I mean, I guess you could do a radio label, and try to get a percentage of it. My feeling is, even though people are saying, well, these neurotransmitters are affecting our mood, I think our mood is affected more by the cytokines being made in the gut.
Mm-hmm.
So if there's a lot of inflammation in the gut, and we're overproducing inflammatory cytokines like TNF-alpha, interleukin-1, interleukin-6. My own personal belief is that that's having more of an impact on mood than the amount of neurotransmitters that are being produced. So that's just my own 2 cents there. But the implication is that when there's less inflammation in the gut, when there's less dysbiosis, the person's mood is going to improve.
Mm-hmm. Great. A quick question. How do you recommend dosing with probiotics, with or without food?
I generally recommend with food.
Mm-hmm. Okay. What do you use as prebiotic?
My favorite prebiotic is arabinogalactan.
Mm-hmm.
You can also use inulin, FOS, things like that. I often find that people get quite a bit of gas from FOS. If you use the short-chain FOS, it's less likely to cause gas, but frankly, I'm one of those people that just doesn't tolerate FOS very well at all. But arabinogalactan is a terrific prebiotic that also has immunomodulatory properties, great data behind it. It dissolves very easily. There's studies giving it to kids. So that, I'd say that's my favorite. I usually give somewhere around a teaspoon a day for most people, up to a tablespoon if necessary.
Mm-hmm. Great. A lot of people really want to know about the types of probiotics you prescribe. Maybe you can talk a little bit about the diversity of species that are available in different blends, things like that.
I have to say that it's highly individual. I could almost say that I give a different probiotic for every patient. I really do. It's because if somebody is generally healthy, then there's some, standard products that have a combination of lactobacilli and bifidobacteria. Mm-hmm. And the most important thing is that they're stable. I've seen products out there that claim to have 100 billion CFUs of the total number of bacteria, but then when you actually count those in a lab, and consumer lab has done this, many of those start dying off as soon as you put them in the bottle. So long-term stability of the product is a really important issue. And so it's important to use a brand that can guarantee that they've got a long shelf life. So if it says that it's got 10 billion CFUs in it, at the time of manufacture, it also should have that many at the time of expiration. So I think if you're talking about a healthy person, you don't really need that many bacteria to get an effect. There's plenty of studies showing you can give somewhere between 1 and 5 billion CFUs of a combination of lactobacilli and bifidobacteria, and that can impact the gut flora. On the other hand, if you're talking about one of these patients that's been in the ICU, where their diversity is totally trashed, I think those people need fairly large numbers of bacteria.
Mm-hmm.
I think they may need 900 billion CFUs a day.
Mm-hmm.
And there's really only one brand that does it. I think most people know who I'm talking about. And that particular brand has got a number of published studies behind it. So I think in that scenario, I'll just say that the brand is VSL number 3. I think that brand, for people who are really severely ill, who have bad colitis, they've got data on it. They have a number of published studies showing that it can help repopulate the gut.
Mm-hmm.
So it really depends on the individual. And there's specific probiotics that have been shown to lower cholesterol. There's some that are better for enhancing immune function. So it kind of depends on what you're going for.
Mm-hmm. And do you see that the products that are marketed for those things really do have the effect that they're marketing for? Because we did have some questions about that too, like how do you tailor probiotic prescribing?
Yeah, it's a tough question. Again, I do a lot of trial and error.
Mm-hmm.
I've kind of been amazed at how some people will respond to a product and others will not at all. I have a patient recently that I was treating who's, he's in Johannesburg, South Africa, and so, I didn't have a lot of control over the products he was getting, but he had pretty bad dysbiosis, and we tried several different products, and he was able to get a hold of an E. coli Distal product, which is a strain of E. coli that's been studied for many years and used for inflammatory bowel disease. And he went on the E. coli Distal, and boom, his symptoms improved dramatically in a very short period of time. So that was clearly the right one for him.
Right.
So I find that I have to do a lot of trial and error with people. I'll try some of the standard brands and see if that works, and if it does, fine. Otherwise, I may go to higher strengths or more diversity or more mixture. I can't say I have found the perfect probiotic, if that's what you're asking, because I think different probiotics work differently for different people. I think we need to do more research. I think when you see the kind of results that people with Clostridium difficile are getting with fecal transplants, which is a 95% cure rate, you have to ask, okay, what is it that is so powerful about these fecal transplants that 1 or 2 infusions can cure a deadly problem? I can't say that that the commercial probiotics that are out there have 95% cure rates.
Right.
They don't, so we've got a ways to go to figure that out there. I think the probiotics that are out there are beneficial. I don't know of a lot of bad probiotics, but they're not giving us 95% cure rates.
Well, and that kind of goes back to at the beginning where you were talking about everyone has their own individual fingerprint. Their microbiome's a different fingerprint, so you might have to try a bunch of different things in different people because you don't really know what's going to work or be effective.
Well, that's why I think doing this testing for not just for pathogens, but to look at the spread of commensal bacteria can give you an idea of what needs to happen in that particular patient.
Right. Well, we have so many good questions, and I wish we could keep you on the line for hours, but unfortunately we need to end our question and answer session there. So again, I'd like to thank you for your time here, and we're just going to go through a couple more slides. So for additional educational materials, we'd like to encourage you all to visit our website, www.gdx.net. On this site, you'll find sample reports, kit instructions, and other information for all of our profiles. In particular, I'd like to bring your attention to Learn GDX. After clicking on the Clinicians tab on the home page, you can access Learn GDX through either the icon you see at the lower portion of your screen or through your My GDX login at the top right. And then once you click on the Learn GDX icon, you'll be taken to this page where you'll see numerous learning modules for your use. These include report reviews, case studies, and in-depth modules focusing on a portion of a test profile. As I mentioned, you can also access this information through your My GDX account. Once logging in, you'll see the online learning link. Once clicking this link, you'll be able to view the Learn GDX modules we just mentioned, as well as gain access to Live GDX, where you'll find a recording of this and previous webinars by clicking on Webinar Archive. And after taking advantage of the materials found on our website, please feel free to contact client services with your questions. You'll see a number on the slide for US and UK customer service. Additionally, please call client services if you need assistance in setting up a MyGDX account. We also offer complimentary appointments with our medical education specialists to answer questions related to our testing, including choosing the right test and reviewing patient test results. Finally, we'd like to encourage you to look for upcoming webinars on our website, www.gdx.net. Next month, we will have Dr. Jill Carnahan speaking on autoimmunity and gut microbes. Thanks again, Dr. Rountree, for a great presentation.
Thank you.